I'm Rhonda Krantz, Program Director at the Water Environment Research Foundation. I recently visited Lake Michigan's North Beach in Racine, Wisconsin. This beach is monitored daily for E. coli by the Health Department, and today it has been deemed safe for swimming and other recreational uses. But did you know that the water sample used to make this decision was collected at noon yesterday? It takes 18 to 24 hours to get a result with the culture-based method currently used. There are new methods that can shorten this time period to as little as 3 hours, enabling a same-day notification decision. In this video, we will provide a brief introduction to one of these methods, QPCR, and take you through some of the major steps involved in its use.
Hi, I'm Julie Kinzelman, and I'm a research scientist. I'm also the lab director at the Racine Health Department and a principal investigator on a WERF project examining pathogen indicators in recreational water. In Racine, Wisconsin, we monitor a variety of aqueous environments: inland recreational waters, coastal recreational waters, stormwater, rivers, and wastewater for the purpose of permitting or the protection of public health. Today we want to talk to you about the evolving science related to water quality assessments, in particular about a rapid method known as quantitative real-time polymerase chain reaction, or QPCR, and the steps necessary to implement this method. QPCR is an analytical method based on quantifying DNA. Proposed revisions to the Beach Act may require coastal recreational waters to be assessed using this method. It would be the first Clean Water Act program to require a molecular standard. In the future, other programs may elect to or be required to use a molecular standard. At that time, agencies will need to make the decision whether or not they want to do this test in-house or outsource it.
I'm here at Quarry Lake Park in the city of Racine. It's a small inland lake that we monitor for the county. The primary uses for this small inland lake are swimming, canoeing, diving, and fishing. As a sampler, one of the first things that I do when I get to a site is I take down information about the environmental conditions. And for taking down those environmental conditions, I record them on a beach sanitary survey form. And this form includes information about the number of bathers, number of waterfowl, the amount of algae present at the site, as well as information about trash and litter on the beach or at the site. One of the important considerations for QPCR is the time factor. And as a sampler, that means that I have to get my sample, put it on ice, and transport it back to the lab as quickly as possible so that it can be processed and results can be provided.
QPCR will provide added benefit for the better protection of public health through more timely notification. But if you are going to perform this procedure in your laboratory, there are some things you need to consider. The first is staffing. Do you have someone on staff who is capable or willing to learn rapid molecular techniques? If no, you can, as we have in our lab, partner with local academic institutions. The second thing to consider is workspace. Not everything can be performed on an open bench, and you will need to have a segregated work area to successfully perform this test. The last thing is the choice of an instrument. There are several instruments available to you with a wide range of costs, and your choice should be based on the number and type of tests you receive and your budget.
Master mix is a set of reagents necessary to perform QPCR. This room has to be kept extremely clean to avoid contamination, so no equipment ever leaves the room. First thing we do is we get the sample out of the refrigerator. This is the filtration apparatus. The sample is added to the funnel where it gets filtered through a polycarbonate filter, which captures everything present in the sample, including the target indicator bacteria. The filter is then folded and placed into the extraction tube containing glass beads. This is our DNA extraction station. Crude extraction is the most basic step. The sample is placed in a bead beater where glass beads mechanically disrupt the cell, releasing the DNA. The sample is is then centrifuged to separate out the liquid containing the DNA for further QPCR analysis. This is the QPCR room where we add the sample to the master mix. The reaction tube containing master mix and sample is then placed in the thermocycler for the QPCR analysis. The final output from this process is to take the data generated by the thermocycler through a series of calculations to express the fecal indicator density as a numerical value.
The science of rapid detection methods is still in its infancy. Considerations such as the suitability of QPCR across multiple water body types still needs to be explored. However, the city of Racine, Wisconsin has used QPCR to quantify E. coli in coastal recreational waters. QPCR will provide people with the rapid notification they so greatly desire and shows promise for applicability in a wide variety of water-based programs. In July of 2010, southeast Wisconsin experienced record rainfall. Communities to the north of Racine made the management decision to close their beaches for a period of 3 to 4 days due to CSO and stormwater events. In Racine, however, the Public Health Laboratory tested our recreational waters using QPCR and made the decision to leave our beaches open. This was the first time we believe that a regulatory action was made using QPCR in the Great Lakes.